Hello, everyone. Today we are taking a look at anaerobic pathways. So far we have talked a lot about aerobic pathways, which required oxygen, as we mentioned before. So what we're looking at in this lesson, lesson number 7, are anaerobic pathways. And we talked about this earlier. Anaerobic means that it does not need oxygen. And in fact, we're going to talk about the fact that instead of oxygen, it might actually use something else. Now, we had previously mentioned in an earlier lesson, without oxygen as a final electron acceptor, do you predict more, less, or the same amount of ATP to be produced? And previously, we had actually seen that without oxygen, there is much less ATP is produced. So we're going to take a look in this course at two different anaerobic pathways. Anaerobic means does not require oxygen. In lesson 6, we talked a lot about the electron transport chain, and we said that ideally we've got oxygen present there at the very end of that, acting as that final electron acceptor after electrons had moved through a whole series of different redox steps. So here it's a little bit different. So the two pathways that we're going to look at in this course is called anaerobic respiration. And essentially what happens there is we still have an electron transport chain, but this time we don't use oxygen as the final electron acceptor. So we use another compound or another thing aside from oxygen. So another compound aside from O2. Not O2, as a final electron acceptor. And we're going to talk about just below there, that next heading, we're going to talk about the fact that another really good final electron acceptor is sulfur. And if you take a quick look at a periodic table, you're going to see that sulfur is actually right underneath of oxygen. So in the same group as oxygen, group 16. So sulfur also has a pretty good electronegativity as well. So more about that to come. We're also going to talk about today is fermentation. And as mentioned in a previous lesson, we've got two sort of examples here, which are lactate fermentation and alcohol fermentation. And again, what all of these things have in common is that they do not require any oxygen. So we're going to look at the details of that shortly. But what we'll start with is anaerobic respiration. And sorry, I'm just noticing here there's a little mistake. We want to make that anaerobic respiration. I'll try to fix that for your notes. So anaerobic respiration. This is a good option for organisms living in low or oxygen-free environments. They can use their own distinct electron transport chain and a compound other than oxygen as a final electron acceptor. So I was mentioning just earlier, a great example of that is sulfur. So sulfur is one example. We also see other organisms might use, maybe I'll just put the chemical formula there. Others might use iron, Fe, and others might use also nitrogen for that. Okay. And as I was alluding to earlier, what makes these particles ideal as final electron acceptors? So thinking back to lesson 6, we discussed the fact that oxygen was a great final electron acceptor because it was the most electronegative in that whole chain of things that happens. So similar concept here. What makes these others, such as sulfur, iron, nitrogen, what makes them really great as final electron acceptors is they have a higher electronegativity than complex 4. because yesterday in lesson 6, we discussed the fact that complex 4 of the complexes would be most electronegative. But we're always looking for the one that has the strongest electronegativity. So these ones that I've mentioned here, they have a higher electronegativity than complex 4, and we talked about this meant that they had a strong pull on electrons. And as I was mentioning earlier, I think a really great kind of tangible example of this is in wetlands where we have something like a rotten egg smell. Some of you might have heard the fact that that's often called like a sulfur smell. And so what's happening in wetlands and places similar to that, like you could even think of like sewage and that kind of stuff, bacteria are producing actually some sulfur byproducts. So here we would have bacteria that use sulfur as a final electron acceptor, release H2S as a waste. Now, interesting about this example and why I really like this example is if we think back to lesson 6, we discussed the fact that in aerobic respiration, in that electron transport chain, we were releasing H2O as a waste product. So here, a little bit different, because oxygen is not that final electron acceptor, now we have H2S instead of H2O that we would have normally seen. So that H2S is as a waste product, and again, that would be like at the end of the electron transport chain. Okay. So in terms of respiration, that's really the one example that I'm going to share with you. Like I said, there's others that belong to like other groups as well. These apply a lot to things that are unicellular. So things like bacteria, things like archaea, even some fungi, etc. But that's really the one example we care about for now for this course. The other example of an anaerobic pathway, which I think is even more interesting to us, is called fermentation. So fermentation, we're going to describe here a little quick recall. It's still going to involve glycolysis, which is something we talked about earlier in lesson number 6. But we're going to see that some other additional things happen. So fermentation, again, just to reiterate, this is also an example of an anaerobic pathway. In other words, a pathway that does not require oxygen. So my first blank over here that I wanted us to remember and recall, glycolysis is a very ancient metabolic pathway that does not require oxygen. And it can occur in basically any cell. So we had discussed in class the fact that glycolysis actually happens in the cytoplasm. So we know that any cell that we could possibly look at, they all have cytoplasm. So glycolysis is able to happen in any of them. Now, in lesson 6, we discussed the fact that during aerobic respiration, NAD plus is regenerated by oxidation, and that happens during the electron transport chain. So we had mentioned that after NADH drops off its electrons and protons at complex 1, that NAD plus can just go back and continue to act as a reactant. So all of these, for example, NAD plus that we see in our diagram, such as here or here, they're all coming from the electron transport chain. So this little diagram down here, it really is just meant to show what happens to those NAD plus. And so they get essentially recycled. So you can see NAD plus over here in glycolysis as well, etc. Also, I missed one down there, but I'll talk about that in a second. So that top half of the diagram, this is all stuff that we learned in lesson 6. Just put that in here briefly. And so that is no surprise to us. Those NAD plus, they are basically recycled. They go back to supply NAD plus needed for these reactions. Because remember, they have to be present there in order to pick up some protons and electrons. Now, the interesting thing is in organisms where this does not happen, so now we're looking at this bottom pathway, if oxygen is not present. we still need a way in order to regenerate that NAD+. Otherwise, glycolysis cannot happen. So in these organisms, they're actually going to use one of two examples, but they're going to use fermentation. And so, as I was just mentioning, the goal of fermentation is to regenerate NAD plus after glycolysis occurs. So, what I'm referring to and what we're going to talk through the steps of, you can see right here in the processes of fermentation, we are actually regenerating this NAD plus so that this can go back and actually help pick up things from glycolysis. That keeps the reactions going. Now, some of you might be thinking, what is the point of doing this fermentation? Why is it so important for that NAD plus to be regenerated? Well, remember from beginning of lesson 6, actually, this NAD plus is needed to make ATP in glycolysis. So I like to think of this from a chemistry perspective. If I'm missing a reactant in chemistry, there's no way for that reaction to continue to go on. We need that NAD plus to be part of glycolysis, otherwise we're not going to get those 2 ATP net that we've been talking about. So the whole goal of all of these fermentation steps that I'm going to show you, I'm starting below and then into the next page, the entire goal is just regeneration of NAD plus. Now, how they differ from one another, because as I mentioned, we've got the 2 different forms. How they differ is the production of different molecules. So we're going to see both examples on the back. We've got lactate fermentation, as I mentioned, and then secondly, we've got the alcohol fermentation. Okay. So some organisms will use these as their primary source of energy, while others, including us humans, can use them as supplemental energy sources when oxygen is limited. So I was mentioning recently with humans, we're going to actually enter that lactate fermentation in times where our bodies are under aerobic stress. So if we're doing some aerobic activity or things of that nature. So I'll explain that further on the back of the page as well. But I wanted to reiterate something that I said earlier. The whole goal of fermentation is to regenerate NAD plus. And the reason for doing that is so that glycolysis can continue. And like I was saying earlier, even though it doesn't seem like very much energy, glycolysis is still making 2 ATP. So let's add that here to make 2 ATP per glucose. So these, especially these smaller organisms, like we're thinking of things that are unicellular, those 2 ATP, that's pretty good for them. Multicellular organisms like you and I, who are doing lots of motion, who have whole like systems, let's call it, for movement and things like that, those 2 ATP are really not going to do too much for us, but again, it's better than nothing. So those processes will help us at least get 2 ATP out of the glucose if we're waiting for some more of oxygen to come in. So let's dive in. Let's actually take a closer look at what these processes are. And again, we're going to see that both of them have the same goal, but really what we get out as a product are going to be very different. So let's start off with the alcohol fermentation. And I put here alcohol slash ethanol. Reason being, chemistry students, you'll know this, ethanol is just the specific name of the alcohol that's formed. And so you'll see ethanol actually in the diagram below. That's not something that we necessarily need to know the picture of in terms of like what we do in this course. But it's just in case, you're wondering, we'll watch kind of the steps going on here. You'll see that ethanol is, it's just a type of alcohol. It's got that OH hydroxyl group on it. Now, alcohol or ethanol fermentation, the types of organisms we would see this commonly used for are things like yeast, bacteria, and archaea. Okay, so again, these are all unicellular organisms. Okay, so humans cannot do alcohol fermentation. Alcohol and ethanol specifically is actually toxic to us. So that's the thing that would be in, and I know you can see the picture there, things like beers and wines, etc. Those are all ethanol, examples of ethanol as well. So these processes are used to actually make those products. So a few steps that I'd like to go over here, and we'll kind of reference that diagram on the left. I've tried to break it down into essentially 3 steps, so similar to what we did with pyruvate oxidation as well. So in step 1, we have glycolysis occurring as normal, so no change about that. We know already that that's going to produce 2 ATP and 2 NADH NH pluses. So this is like step 1. This is just our glycolysis step. So splitting up glucose into pyruvate. Now, instead of entering pyruvate oxidation, in step 2, what's going to happen instead is really fermentation. So I'm going to make that little arrow here, step number 2. And what we see happening here is removal of carbon dioxide. And we actually had some vocabulary in order to describe this before. We called it a decarboxylation. So we've got a decarboxylation reaction resulting in the removal of CO2 gas, carbon dioxide gas, from pyruvate. So this step, we also saw a similar step happening in pyruvate oxidation, but the rest of that is actually going to be different. So let's just highlight for ourselves because I think it's maybe helpful. We can see this carboxyl group right here. This is going to be actually removed as the 2CO2. Okay. We mentioned this before. For fermentation to happen from 1 glucose, you can probably notice there's some 2's out in front here. So glucose, we know, splits into 2 pyruvate, and then each pyruvate would go through this set of steps, producing a total of 2 carbon dioxide. In step 3, this is where something brand new is going to happen. So I'm going to put in right here step 3. This is sort of like this arrow, this kind of area here. And what happens in step 3, this acetaldehyde, which is like an intermediate product, it is going to be reduced by NADH to form NAD+, and as well ethanol. So ethanol is that last product that you see at the very end there. And so here's where I really wanted to reinforce something that I was talking about earlier. Notice here the NADH right here, it's adding its electrons because remember, it's an electron carrier. And where it's adding its electrons are actually over here. So we see actually that suddenly an H comes here and we've got also an H that comes here. So it's actually added to this molecule some of those hydrogens that we were talking about previously. And in doing that, my whole point about this whole thing and what the goal was, we have allowed regeneration of NAD+. And we said NAD+ is needed for glycolysis. We know that's going to produce for us this 2-NADH, and that cycle can continue. So as we said, with aerobic respiration, that NAD+ comes from the electron transport chain. But if you don't have oxygen present and you have no other electron acceptor, this is the best you can do with cycling that NAD+. So making sure that NAD+ is present and available for glycolysis. So what that brings us to is to sort of think about a little bit is what an overall sort of, reaction for this would be, which might also include glycolysis. So let's talk about first. We know with glycolysis, we start off with glucose. So I'm going to just write glucose in here first. And we also talked about the fact that in order for glucose to actually make pyruvate molecules, it needs to have NAD plus present for that. So I'm actually going to write in an NAD plus here. I know we don't normally, but I think it will help to actually, trace the pattern as to what's happening. So let's put in here 2 NAD plus as a reactant just to show that, we need that for the reaction to actually get started. Now, what this is going to form as a final product, because we know in between there's 2 pyruvate and that's fine, but we also want to think about like what our overall product is at the end. So I was talking about this previously that, one nice way to do this or one, simple way to do this is talk about what compound at the end still has carbon. So I'm going to make a little arrow here. This is, like I said, a simplified, type of diagram here. I'm going to actually first do my pyruvate oxidation. So right now, glucose NAD+, we're going to actually produce 2 of pyruvate. We're going to produce also 2 ATP. And like we were just talking about, we'll produce 2 NADH. So this whole thing that I'm writing, this represents glycolysis. Okay, so similar to what we've talked about in lesson 6 already. But now we want to actually regenerate this NAD+. So at this point, just doing glycolysis, we have not regenerated anything. So what we're doing in that sort of second step is that we're actually taking a look now at how do we get that NAD+ back. So what we're going to show now is how we get from essentially pyruvate all the way to ethanol. So again, we've got 2 pyruvate that are going to start that reaction. Okay, we're going to see that those 2 NADHs are used. Okay, and then we're going to essentially release or have as a final product. Well, we know there's 2 CO2 there. That's one place where carbon goes. And another place is the 2 ethanol. Let's just write that as words for now. And then, as I was saying, the whole goal of this is that in this arrow over here, we see regeneration of NAD+, which was our whole goal of doing fermentation. Okay, so point about all of this, and this would be what I would call the fermentation steps. So let's put here alcohol fermentation, is that we are yielding or we are actually producing again the NAD+ needed here. So this is very much a cyclical thing, producing NAD+ in order to use that to actually do glycolysis all over again. Now, I wanted to mention something briefly with here, now that we've kind of understood a little bit about how the cycle goes. I want to talk about some human applications, and I know some of us have talked about this previously as well. Why would people care about this? Because this doesn't happen in humans. Well, the reality is we've got some sort of like economical and like sort of functional things that we use this for. And I want to point out to you guys these 2 products, 2 CO2 and 2 ethanol. So let's actually talk about this for just a second. The ethanol, as I said earlier, ethanol is a type of alcohol. Okay, and we can actually use that in alcoholic drinks, for example. So that's just shown on the side there. But the second one, which is my favorite one, is what we use it in terms of baking and bread. So the CO2 that is produced by this, the CO2 is that air that allows our dough to rise. So I'm going to mention right here, production of CO2 gas okay, this is just production from fermentation, allows dough to rise. Okay, so some of you might be wondering, well, wait a second, if that's what's happening in my bread, why doesn't my bread have alcohol in it? And some of you might already know this from chemistry. Ethanol has a very, very low boiling point. So when we bake the bread, we actually do essentially evaporate off any of the ethanol that has formed. So when we're baking the bread, we're actually kind of speeding up the reaction. We're warming things up, right? Thinking of particle theory. And in doing that, there's little bubbles of CO2 gas that actually get trapped in there, and that allows our dough to actually rise. And as I mentioned, the ethanol part, because we're baking it, it's actually going to evaporate off very easily. Ethanol has a very, very low boiling point. So even though some ethanol is made, it's all going to be evaporated off. So those are some sort of like, I guess, tangible human examples, like what we use fermentation for. Now we're going to take a look at another one called lactate fermentation. Again, as I was saying earlier, a common thread between any type of fermentation, our goal is always regeneration of NAD plus so that it can be used again for glycolysis. So we're going to actually see that with lactate fermentation. Big difference is instead of making alcohol, we're going to be making lactate, which is something different. So I mentioned right here that this is used as a primary energy source in many microorganisms. So thinking of things like, again, bacteria and archaea, okay, those sort of like really, really small, sometimes unicellular ones. But it's interesting too that we can also use this in eukaryotic cells. And I was mentioning that human muscle cells actually do use this type of fermentation. And the sort of examples where this would happen is when ATP demand exceeds the rate at which oxygen can be supplied to the electron transport chain in the mitochondria. So this would be things like aerobic activity. So we know, and I'm sure all of us have experienced this, but we also learned this in grade 11 biology, is when we are doing these strenuous sort of aerobic activities, our heart rate and our breathing rate increases. And the goal of that is to get as much oxygen to our cells as is possible. However, there does come the point where not enough oxygen gets there fast enough. So that's what I'm describing here is oxygen does not get to our cells fast enough. So we're doing so much activity that essentially all the oxygen is in use and we're just waiting for more to actually get into those cells. So as I mentioned here, during strenuous aerobic activity, glycolysis will happen rapidly and it will still produce as much ATP as it can, right? So still it's going to be making those 2 ATP per glucose. However, we still have that problem of NAD plus. We still need that to be regenerated really quickly in order to keep things going. So we still are going to have that process of pyruvate oxidation like we were talking about before. We're still going to have the process of glycolysis like we had before. But what we see next is that pyruvate oxidizes NADH and it's converted directly into lactate, which you can see on this diagram down here, that's called lactate. And so similar to before, in this process, we are going to actually reproduce or regenerate, like we said, NAD+. So that's our entire goal. Now, it says here, when oxygen levels return to normal, the reaction can be reversed to produce pyruvate again, which will continue onto pyruvate oxidation, citric acid cycle, and the electron transport chain. One thing I'll mention about this one, because if you're Googling it or you're reading up on it, occasionally instead of seeing this as lactate, you will see this called lactic acid. And the reason for that is that if you notice there's that carboxyl group at the bottom, that COO minus. Remember from earlier in the course, we said carboxyl is unique in that it could actually gain a proton or lose a proton. So in this case, COO could actually be COOH. It could actually gain that proton back. And so that is an acidic proton. That's a proton that is easily lost by lactate. So for our purposes, you can say either of those things. You can call it either lactate as a product or lactic acid. Both of them are technically true. Now, one thing interesting about this, which is sort of a misconception, some of you might have heard about in your travels that lactic acid builds up in our cells and that causes us to have like muscle pain. It's since been shown actually that the muscle pain that we get, it's actually from the tearing of our muscle fibers and not from lactic acid buildup. So lactic acid, although it might be produced, our body clears that away pretty quickly because, like I said, often it can make it back into pyruvate. But what the kind of misconception of this is that the pain that we feel in our muscles is actually from lactic acid. In fact, if you look into it a little further, it's actually from our muscles tearing and then regenerating themselves. So less so from this lactic acid product and more from actually this sort of regeneration part. Now, what I'd like to mention here is I do have a couple homework questions down at the bottom. I've also linked into our daily slide deck, which I'll put with this video, is just another sort of fermentation video to take a look at. As you all know, this will be the last lesson that goes towards our cellular respiration test. So as you've looked through the video and as you've looked through the lesson notes that I've left you, I'm going to ask you guys just to jot any questions about this down that you have, and then hopefully tomorrow when I'm back, we can actually go through and talk and answer any of those questions.